Hi everyone, I'm Jess Rudd. I'm a graduate student from the University of Exeter in the UK, collecting data here at CEI for my master's, looking at how to use novel tracking techniques for juvenile green turtles. Incidentally, tomorrow is Endangered Species Day, so the data that we've been collecting as a team is going to be contributing to the knowledge of green turtles, which are endangered species.
Hi, good morning everybody. My name is Christian Danielle and I am the other advisor for the turtle research class. I also work as a shark and ray conservation research technician over at CEI. But without any further ado, it is my great pleasure to introduce you to the turtle team.
Good morning and welcome to Turtle Team. My name is Emmett Canner.
I'm Keenan McMorrow.
I'm Brad Stanton.
I'm Julia Foreman. I'm Megan Long.
I'm Michael Freeman.
And I'm Ellie Adams.
We conducted our research on the homing behavior displayed by the juvenile green sea turtle, or Colonia mitis, with special attention to habitat quality. Many animals display homing behavior. Oh, there we go. Awesome. Many animals display homing behavior and have the ability to travel incredible distances with remarkable accuracy. For example, salmon returning from the ocean to spawn can pinpoint the exact stream where they are born. And many migratory birds return to the same stopover sites year after year. Chemical cues, physical landmarks, and magnetic fields can all be used as guides to aid in these animals' migrations. Turtles rely primarily on geomagnetic orientation, or positioning using the Earth's magnetic fields by detecting tiny variations in the intensity and inclination angles of these fields through a process not fully understood by science. They establish very specific home ranges and have demonstrated the ability to return to these ranges with remarkable consistency.
Green sea turtles are ecosystem modifiers due to the fact that their grazing habits improve the seagrass quality and also help to increase the habitat quality for commercially significant species such as conch, grouper, and juvenile fish. Their feeding or grazing also helps to strengthen the roots of the seagrass, and which this helps with erosion. So, threats to turtle populations. Green sea turtles are an endangered species, and there are several threats to this. The first one is habitat loss, which can be oil spills or coastal development. Coastal development would be the removal of seagrass. Next would be anthropogenic threats, which are human threats, and that can be bycatch, which is the accidental catch of a non-target species, and also oil spills and coastal development, as I said earlier. And finally, low egg survival rates can be attributed to the above-mentioned threats. Finally, the Bahamas, the Bahamian government has put in place a ban on all turtle catching and trading since 2009 in order to help to increase the populations.
Primary studies on green sea turtles have been on adult female turtles. This is because year after year, like clockwork, female turtles return to their natal beaches to lay eggs. This gives an opportunity for scientists to record data and learn about their homing behaviors. Some turtles have shown the ability to relocate their capture sites from over 130 kilometers away in a year and a half in captivity. On the other hand, juvenile green sea turtles are data deficient, meaning there hasn't been much studies done on them. This gap in knowledge has sparked the interest in our research. Our research objectives are to study the homing behavior and movement patterns of juvenile green sea turtles. The turtles that we're studying primarily live in Starved and Poison Creek here in southern Eleuthera. We're doing this to understand the relationship between homing ability and foraging habitats for juvenile green sea turtles.
So our study was conducted in two shallow tidal creeks named Poison Creek and Starved Creek, located in southern Eleuthera, and these two creeks are located 4 kilometers apart from one another. On the map, you can see that Poison Creek is on the left side and Starved Creek is on the right side. And the reason for habitat mapping was that we wanted to assess the quality of the habitat by looking at the percentage of the seagrass and macroalgae sediment type as well as the depth. And the reason for this was that we wanted to see if the juvenile green sea turtles movement and the use of space within the creeks, and if they preferred one creek over the other after we displaced them back into the water. And lastly, why we chose these two sites was because the turtle research team at CEI has been doing long-term population monitoring over the past 5 years, so we know that the turtles within these creeks have high site fidelity.
So the method we use to catch the turtles is called the rodeo method. And what we do is that we will go on the boat and look for turtles. Once we spot one, we will follow it. While we follow them on the boat, we tire them down. And once they're tired enough, we will send snorkelers in. Then they will catch the turtles and then move them onto the boat, and we will later displace them into a different creek. Then on the boat, we measure the turtles. What we do is we measure the head and the carapace, and then we also weigh them. Lastly, we attach tags. The first tag we attach is the GPS tracker. This is attached to the back of the carapace of the turtle, and it's attached to a 4-meter long tether. This allows the GPS to stay afloat and connect with satellite. The GPS tracker also gives us every motion of the turtle. The identification tag is on the back 2 flippers of the turtle. This lets us identify them once we catch them again. And lastly, we attach an accelerometer. This tells us the depth, temperature, acceleration, and magnetic fields, and 3D movement.
So far as Julia described, we have successfully mapped both the Starved and Poison Creek. And as you can see by the dots, the larger the dots and the more darker red, those represent areas of higher seagrass abundance. And so after we mapped each of the habitats, we have started capturing turtles. And we've captured a total of 10 turtles. 5 of those we left as controls, and 5 of them we displaced from one creek to the other to test their homing behaviors. And of the control turtles, we recovered the GPS data of 2 of them, and they did pretty much as we had hypothesized. They stayed each in their relatively small home ranges and frequented areas of high seagrass abundance. For the displaced turtles, we recovered the tracking data of 2 turtles that we displaced from Starved Creek to Poison Creek. And of those 2 turtles, one of them returned home, and one of them actually remained in Poison Creek for up to 4 days that we tracked it. And this was surprising to us, but we also inferred that it may have returned back home starved and just taken longer than 4 days. And in the future, we'd like to track them for longer to see how they react. For the last turtle that we recovered the data from, we displaced it from Poison Creek to Starved Creek. And as you can see, it did eventually return home after spending about 2.5 days exploring its environment in Starved Creek. And one significant observation that I found is that all of the displaced turtles spent significantly more time exploring their new environment and attempting to orientate themselves than they actually spent returning home. So for example, this turtle spent about two and a half days in its new environment, and then just a few hours returning over to Poison Creek. Lastly, using the accelerometer data we recovered, we created a graph comparing the movement speeds of the control versus the displaced turtles, and we found that the displaced turtles on average moved about twice as fast and covered twice the distance than the control turtles, and we attributed that to the fact that they were most likely exploring their new environment and attempting to orientate themselves. And one discovery that we found surprising was we used an ANOVA test to compare all this data, and we found that the displaced turtles, there was no significant difference in their movement speeds during the daytime versus nighttime, whereas the control turtles were definitely much more active during the daytime with feeding and rested more at night.
Based on our results, we discovered that the displaced turtles are much more active than the control turtles. We can attribute this to them orientating themselves in this unfamiliar area, whereas the control turtles already are familiar with the area that they are placed in and know where the seagrass is to feed. This supports the past studies that have been done on the adult turtles and is what we were expecting to find. Additionally, we found that 50% of the displaced turtles returned home, which corresponds with the past studies on adults that have been done, and it took at most 3 days for the turtle to return home, which is relatively quickly. Past studies of relocation have most been done on smaller organisms, so relocation on a larger migratory species has been less researched. Based on our results, we have also discovered that relocation is not a long-term conservation method for juvenile green sea turtles, so we need to look to the habitats in which they already inhabit and conserving those because the turtles will continue to return home to these ecosystems even if they are toxic. However, we now know that if a turtle is needed to be kept in captivity or rehabilitated, that when placed back into the ocean, it will be able to find its way home no matter how long it's been in captivity or the distance it has to travel. Lastly, when looking to the future in future studies for juvenile green sea turtles, we would like to track the turtles for longer so we can see that if over time the turtles that didn't return home will return to home, and once they're home, if they can stay there. We would like to thank our advisors, Jess and Christian, and we would also like to thank the rest of the turtle team, Megan, Haley, Chelsea, Annabelle, along with the Island School, Cape Eleuthera Institute, and University of Exeter. So the question was, are the turtles that we have tracked so far still wearing all their gear? So as we mentioned, a lot of the turtles we haven't successfully been continued to track them because we've either lost the tracker or it's fallen off, but a lot of them still wear the accelerometer, but when we go out and recollect the turtles, we take off the accelerometer to keep all the data, so a lot of the turtles don't have their trackers on anymore.
So you have to redo to lengthen stuff.
Exactly, yeah.
Did you say there was a tether that was attached to the turtle, which is floating on the surface for monitoring? And if so, does that get snagged on coral? How often? So forth.
The question was about the GPS tracker that we have and the tether that is attached to the turtle. And yes, there's a tether that is attached to it so it can stay on the surface and we can collect the data. However, there is a part of the tracker that it has a 30-pound pull, so if it does get caught in anything, the turtle will be able to get loose and won't get caught up with it. Yeah, you.
What effect do accelerometers have on sort of movement of turtles?
The question was, what does the effect of accelerometers have on the turtle?
So anytime you're taking a wild animal out of its habitat and modifying it, adding data collection, tethers, all of these instruments that we use to collect scientific data, you're not going to be benefiting that animal. So it's probably fair to assume that putting this accelerometer on their back, it's going to decrease their streamline movement through the water. We might not be able to move as quickly, but the effects on the turtle we hypothesize are minimal. And furthermore, one of the hopes of our study is that even though we may have to inconvenience a couple turtles, the good that we're going to be producing for the greater species from our study will outweigh those negatives.
So there's such things as like a turtle hatchery, and are those effective at helping endangered status?
So the question was, are there things such as turtle hatcheries and whether or not they are effective in protecting turtles?
In some places there are protected beaches where people will allow turtles to come up and lay their eggs in a protected place where other animals cannot go and compromise the eggs. and will allow them to hatch naturally and go into the ocean. I don't believe that it is possible to have a turtle hatchery because turtles need to spend much of their life out in the open, and they have many stages to their lives. When they're first hatchlings, they escape into the ocean, find some pieces of seaweed, and they float with it and hide for protection. And if we wanted to release the turtles after captivity, if we wanted to raise them for a certain amount of time and then release them in captivity, they really wouldn't know how to escape from predators or find good grazing habitats, or even really, as our study is about homing behavior, they might not know how to use their homing behavior as well.
Just to add on to that, there are turtle hospitals, and for the very beginning stages of a turtle's life cycle, before it has to do all the things that Brad mentioned, in learning how to survive in the wild, there are centers that specialize in hatching the turtles and then releasing them to the wild. So hospitals and basic hatcheries are effective conservation tools.